requires a certain mindset. They really push the boundaries of what we even consider life. Fundamentally, they're just information genetic code wrapped in a protective layer, utterly dependent on hijacking the machinery of living cells. Okay, so let's start with the basics. What is a virus? At its core, it's unbelievably minimalist. You have the genome, that's the genetic blueprint, and this genome, it can be DNA or RNA. Crucially, it might be single-stranded, SS, or double-stranded, Ds, and it can be linear, like a piece of string or circular. This genome is protected by a protein shell, the capsid, and often that's pretty much it.
So blueprint and suitcase, simple yet effective, I guess. But what do these components actually do? How do they let the virus survive outside a cell?
Well, the genome is straightforward. It contains the instructions. The code for making viral proteins, the signals needed to take over the host cell. The capsid is more complex. It has two main jobs. First, protect that fragile genome when the virus is floating around. Second, it often has structures on its surface that help it stick or attach to the right host cell. And these capsids, they aren't just random blobs. They form specific, highly organized geometric shapes. You see icosahedral ones, like a 20-sided die, very strong, or helical, like a rod. And sometimes, especially with viruses that infect bacteria, you see really complex combinations of shapes.
That makes sense for protection. But then there's this other layer. Some viruses have the envelope. What's the deal with that? Why add complexity?
Ah, the envelope. That's a really clever bit of biological trickery, almost like camouflage, the lipid membrane, essentially fat. But here's the key. The virus steals this membrane directly from the host cell as it leaves. It's like wrapping itself in the enemy's flag. Studded within this stolen membrane are viral proteins, specifically glycoproteins. These are vital. They're the keys the virus uses to recognize and then fuse with the next host cell.
Okay, so stealing the membrane gives it an advantage. But does it create any weaknesses?
Absolutely. This leads to a fundamental split. We talk about naked viruses, just the capsid and genome and enveloped viruses. Naked viruses like, say, poliovirus or adenovirus are tough. They can survive disinfectants, stomach acid, drying out on surfaces. They're built to last. Enveloped viruses, think HIV, influenza, SARS-CoV-2, are much more fragile outside a host. Soap, heat, detergents easily disrupt that fatty envelope. But that envelope gives them advantages too. It helps them enter cells more easily through fusion, and it can help them hide from the immune system. Stability versus stealth, basically.
Got it. And besides these core parts, the notes mention accessory enzymes. Why would a virus carry its own tools if it's just going to use the host stuff?
Because sometimes the host cell simply doesn't have the tools the virus needs for its specific strategy. Think about it. Our cells replicate DNA, right? But they don't usually replicate RNA from an RNA template. So RNA viruses often need to pack their own RNA polymerase. Retroviruses are a classic example. They must carry reverse transcriptase to turn their RNA genome into DNA. Others might pack proteases to chop up viral proteins or integrases to splice viral DNA into the host genome. These aren't optional extras. They're essential for their particular life cycle.
This diversity DNA, RNA, single, double-stranded, plus these extra enzymes make simple classification tricky, which is why the source material really emphasizes the Baltimore system. Can you unpack that? Why is how they make mRNA the key?
Right. The central dogma, DNA to RNA to protein. All viruses, no matter what their genome is made of, eventually need to make messenger RNA, or mRNA. Why? Because that's the language the host cell protein-making factories, the ribosomes, understand. Yeah. The Baltimore system, developed by David Baltimore, brilliantly classifies viruses into 7 groups based purely on their genome type and the pathway they use to generate that essential mRNA. It gives us a logical framework.
Okay, 7 paths to making protein instructions. Let's walk through them.
Class I are the dsDNA viruses, double-stranded DNA. Examples are adenovirus, herpes viruses. These guys are pretty conventional. They mostly use the host cell's own
Let's
enzymes,
work today
the
to
ones in the
make
nucleus,
tomorrow a
to
better
transcribe
day.
their DNA into
Okay.
mRNA. They sort of follow the
You
host
can
playbook.
do
So
it.
they fit in
Let's
relatively
work
easily.
today to
Exactly.
make
Class
tomorrow a
2
better
are ssDNA
day.
viruses,
Okay.
single-stranded DNA, like
You
parvovirus.
can do it.
Now, the host
Let's-
machinery is designed for double-stranded DNA. So these viruses have an extra step. They first have to synthesize a complementary strand to turn their genome into a double-stranded form. Then the host machinery can transcribe it into mRNA.
Makes sense. What about the RNA viruses?
Okay, now it gets more interesting. Class 3 are dsRNA viruses, double-stranded RNA, like reovirus.
Yeah.
Host cells don't have enzymes to read double-stranded RNA. So these viruses must package their own viral RNA polymerase right inside the capsid. As soon as they enter the cell, that enzyme gets to work, making mRNA from the dsRNA template.
And then the single-stranded RNA viruses. The notes stress that polarity positive, plus, or negative sense is everything here.
Absolutely critical. Think of polarity like the direction the code is written in. Class 4 are positive sense, single-stranded RNA, plus ssRNA viruses.
Hmm.
Poliovirus, SARS-CoV-2 are key examples. Here, the plus RNA genome has the same sequence as mRNA. It's message ready. As soon as it enters the cytoplasm, the host ribosomes can grab it and start translating it directly into protein. It's incredibly efficient.
Like a pre-written message.
Precisely. Now, contrast that with class V, the negative sense single-stranded RNA, ssRNA viruses. Think influenza, rabies. Their genome is the complementary strand mRNA. It's like having the photographic negative. You can't read it directly. So just like class 3, these viruses must carry their own viral RNA polymerase with them. This enzyme first makes a positive sense copy from the negative sense template, and that positive strand then serves as the mRNA for translation.
Okay, so classes I through V cover a lot of ground just getting to the mRNA stage. But the last two, MIND 7, do something even weirder involving reverse transcription, right? Going against the usual flow of information.
Exactly. They break the standard DNA to RNA rule. Class 6 are the famous retroviruses, with HIV as the textbook example. They have a plus ssRNA genome. But crucially, they don't use it directly as mRNA initially. Instead, they use their packaged enzyme, reverse transcriptase, to make a DNA copy of their RNA genome. This DNA copy, called the provirus, then gets integrated into the host cell's own chromosomes using another viral enzyme, integrase, becomes a permanent part of the host DNA.
Wow, that's a long-term commitment.
It is. And finally, class 7. These are technically dsDNA viruses like hepatitis B, but they have a peculiar replication cycle that involves an RNA intermediate. They make an RNA copy of their DNA genome and then use reverse transcriptase to convert that RNA back into DNA to make new viral genomes. It's a sort of DNA loop. So the Baltimore system isn't just classification, it's a functional roadmap. It tells us what enzymatic steps are crucial for each virus type, and importantly, where we might be able to intervene with antiviral drugs.
Okay, that framework is super helpful. It highlights that despite all this structural and genomic variety, every single virus operates under the same principle. Molecular parasitism. They have to hijack the host. And the most critical point, which dictates their entire strategy, as you mentioned, is their lack of ribosomes. No protein factories. DNA, RNA, positive, negative, doesn't matter. They must take over the host translation system. This dependency forces a very specific sequence of events for infection.
It's like a heist, perfectly choreographed. 7 distinct steps. If any single step fails, the whole operation collapses. It's a beautiful, if destructive, display of biochemical necessity.
Let's walk through this heist stage by stage, focusing on the molecular details the notes provide. Starting with the first contact. Step 1. Attachment. This really is where structure defines infection. It's a specific lock and key mechanism. The virus isn't just randomly bumping into cells. It has proteins on its surface that bind tightly to specific host receptor proteins on the cell membrane. This explains why viruses are often species-specific or even tissue-specific. The notes give great examples. HIV needs to bind not just to the CD4 receptor on T helper cells, but also to a co-receptor, usually CCR5. No CD4 and CCR5, no infection of that cell.
Right. Or influenza, which targets specific sialic acid sugars found on proteins coating the cells lining our respiratory tract. The virus is essentially checking the cell's ID badge. If the badge isn't right, it can't get in.
Step 2. Entry. Okay, it's attached. Now it has to get inside. How does that happen?
2 main ways, depending on whether it's enveloped or naked. Enveloped viruses often use membrane fusion. Those viral glycoproteins we mentioned earlier rearrange and basically merge the viral envelope with the host cell membrane, kind of like 2 soap bubbles fusing. This dumps the inner part of the virus, the nucleocapsid, directly into the cell's cytoplasm. Naked viruses can't fuse, so they often trick the cell into engulfing them through a process called endocytosis. The cell membrane wraps around the virus particle, pulling it inside within a little bubble or vesicle called an endosome.
Step 3. Uncoating. So now it's inside, but it's still trapped in its protein coat, the capsid. How does it get the genetic material out?
This uncoating step is crucial. The capsid needs to break apart. Often, it's triggered by changes in the environment inside the cell. For viruses that enter via endocytosis, remember they're inside that endosome bubble. Well, the cell naturally makes the inside of endosomes more acidic over time. That drop in pH can cause the capsid proteins to change shape and fall apart, releasing the viral genome into the cytoplasm where it can finally start working. Sometimes specific host enzymes also help break down the capsid.
That feels like the real point of invasion. The genome is loose.
It really is the point of no return. The virus is now committed.
Step 4, replication and transcription. This is where the Baltimore class dictates everything. The virus needs to make copies of its genome and make mRNA to produce proteins.
Exactly. Depending on the class, it might use the host's DNA polymerase in the nucleus, class 1, or its own reverse transcripts, class 6, or its own packaged RNA polymerase, class 3. The goal is twofold: replicate the blueprint and transcribe the messages.
Step 5, translation. Now the hijack is complete. The host ribosomes get commandeered. They start reading the viral mRNA and churning out viral proteins.
And for many viruses, especially complex RNA viruses like polio or coronaviruses, there's another efficiency trick. They often make one single gigantic protein strand called a polyprotein. This long chain contains all the different viral proteins needed, linked end to end.
But that long chain isn't functional yet, right?
Correct. It needs to be chopped up into individual functional proteins. And that chopping is done by specific viral enzymes called proteases. They act like molecular scissors, cutting the polyprotein at precise locations.
And that's why protease inhibitors are such effective drugs, like for HIV.
Precisely. Block the protease, the polyprotein doesn't get cleaved, the virus can't assemble functional parts, and the replication cycle grinds to a halt.
Step 6, assembly. Okay, so now we have new viral genomes and all the necessary proteins, structural ones for the capsid, enzymes, etc. How do they all come together?
Remarkably, this assembly process is often spontaneous. The protein subunits and the genome are designed to self-assemble, like puzzle pieces clicking together, driven by their shapes and chemical properties. Sometimes, temporary helper proteins called scaffold proteins are involved. They guide the proper folding and construction of the capsid, and then they're removed before the final virus particle is complete. It's incredibly efficient molecular construction.
And the final step, getting out to infect more cells. Step 7, release.
Again, depends on naked versus enveloped. Naked viruses generally don't care about the host cell membrane. They often accumulate in huge numbers inside the cell until it simply bursts open, a process called lysis. This kills the cell, but releases thousands of new viruses all at once. Enveloped viruses need that host membrane. So they exit via budding. They push outwards through the cell surface, wrapping themselves in a patch of the host membrane, studded with those viral glycoproteins as they go. This budding process can be slower and doesn't necessarily kill the cell immediately, which can lead to chronic persistent infections. Right, that 7-step cycle describes the typical acute infection. The virus gets in, its copies gets out, often destroying the cell. But as the study notes really emphasize, some of the most successful and, frankly, most insidious viruses have another strategy. They hide. They establish latency.
Latency, the stealth mode. It seems like a brilliant evolutionary tactic, especially if the host has a strong immune system that would otherwise clear the infection quickly. How do they manage to stay hidden?
There are two main ways they stash their genetic material long-term. The first is integration, becoming a permanent part of the host's own DNA. This is the hallmark of retroviruses, like HIV. After reverse transcription creates that DNA provirus, the viral enzyme integrase literally cuts the host chromosome and pastes the proviral DNA in. Specific sequences on the viral DNA, called LTRs, long terminal repeats, help guide this process. Once integrated, the viral DNA just sits there. Every time the host cell divides, the provirus gets copied along with the host's own genes. It's passed down to all daughter cells, essentially becoming part of the cell lineage, waiting for a signal to reactivate.
So the virus becomes a hidden passenger, inherited by the cell's descendants. What's the other main hiding strategy?
That's episomal latency. Instead of integrating into the chromosome, the viral genome persists as a separate piece of DNA, usually circular, called an episome. This is typical of herpes viruses, think herpes simplex, cold sores, Epstein-Barr virus, mono, cytomegalovirus. These episomes often hang out in long-lived cells like neurons or certain immune cells, B cells. During latency, they express very few, if any, viral genes, making them almost invisible to the immune system. Reactivation can happen later, often triggered by things like stress, illness, sunlight exposure, or immunosuppression. The episome then wakes up and starts the lytic cycle again, causing recurrent symptoms like that cold sore appearing.
It's fascinating how these different families evolve distinct long-term survival plans.
And what's truly amazing is how ancient this strategy is. The notes point to bacteriophage, lambda, a virus that infects bacteria. It does almost the same thing as herpes viruses, but with integration. Lambda uses an enzyme called recombinase to splice its DNA into the bacterial chromosome, where it sits dormant as a prophage. It stays quiet until the bacterium gets stressed, maybe DNA damage from UV light or starvation. That stress triggers excision enzymes that cut the prophage back out, initiating a massive burst of virus production, the lytic cycle. This hiding strategy is billions of years old.
Wow. But sometimes the virus doesn't just hide, it fundamentally corrupts the host cell, pushing it towards cancer. This idea of host cell transformation and oncogenesis.
Yes, and the source material makes a crucial historical point here. Studying how viruses cause cancer was absolutely pivotal. It basically taught scientists the fundamentals of how genes control cell growth and how that control can go wrong. Before viral oncology, cancer mechanisms were largely a black box.
So how does a tiny viral blueprint manage to derail something as complex as the cell cycle and cause uncontrolled growth?
Generally, two main ways. Sometimes the virus itself carries a cancer-causing gene, a viral oncogene, that directly stimulates growth pathways. More commonly, though, it's about interference. The second way is insertional mutagenesis. When a virus like a retrovirus integrates its DNA into the host genome, where it lands can be random. If it happens to land in or near a host gene that regulates growth, it can either accidentally switch on a growth-promoting gene, a proto-oncogene, or, more often, disable a crucial tumor suppressor gene, one of the cell's natural brakes.
Let's look at the specific examples in the notes. HPV seems like a prime case.
Human papillomavirus, particularly high-risk types like 16 and 18, is a classic example of viral interference. HPV makes two key proteins, E6 and E7. To understand their impact, you need to know about two critical guardian proteins in our cells, P53 and RB. P53 is often called the guardian of the genome. If DNA gets damaged, P53 tries to halt the cell cycle for repairs, or if the damage is too severe, it triggers programmed cell death, apoptosis. RB retinoblastoma protein is a major gatekeeper for cell division. It keeps the cell cycle paused until everything is ready.
So E6 and E7 are like targeted missiles against these guardians.
Exactly. E6 targets P53 for destruction. E7 binds to and it activates RB. By taking out both the emergency brake, P53, and the main cell cycle checkpoint, RB, HPV essentially forces the cell to keep dividing, even if its DNA is damaged and accumulating mutations. This uncontrolled proliferation is what leads to cervical cancer.
Are other viruses using similar tactics on different pathways?
Yes. Take EBV, Epstein-Barr virus, linked to Burkitt lymphoma and other cancers. EBV produces a protein called LMP1. LMP1 mimics a permanently switched-on cell surface receptor. This mimicry constantly activates a signaling pathway inside the cell called NF-κB. NF-κ normally promotes inflammation, cell survival, and proliferation, but it's tightly regulated. EBV through LMP1 just keeps that pathway blazing, promoting the survival and growth needed for lymphoma development.
Interesting. The notes also mention HBV and HCV causing liver cancer, but suggest it might be less direct.
That's right. Hepatitis B and hepatitis C viruses are major causes of hepatocellular carcinoma, liver cancer. But it seems less about specific viral oncogenes like E6, E7, or LMP1, and more about the consequences of chronic infection. Years, often decades, of persistent infection lead to continuous liver damage, inflammation, cell death, and regeneration. This constant cycle of destruction and repair massively increases the chance of random DNA mutations accumulating in liver cells over time, eventually leading to cancer. It's oncogenesis driven by chronic inflammation.
And the last example, the retrovirus HTLV1.
HTLV1, human T-cell lymphotropic virus type 1, is different again. It integrates into the DNA of T cells, a type of immune cell. It produces a protein called TAX. TAX is a potent oncoprotein that directly interferes with cell cycle control and signaling pathways within the T cell, essentially forcing the infected T cells to proliferate uncontrollably. This leads to a specific type of cancer called adult T-cell leukemia lymphoma. In every case, studying the virus revealed critical control points within the cell. Viruses were like molecular probes that showed us the switches for life, death, and uncontrolled growth.
Okay, we've gone from the single-cell hijack to long-term consequences like cancer. Now let's pull back that lens, use the telescope view the note suggested. The argument here is really provocative. Viruses aren't just pathogens, they're genetic couriers, evolutionary editors. They actively shape biodiversity across the tree of life.
It's a fundamental shift in perspective. They become agents of change, not just destruction. They are major drivers of horizontal gene transfer, HTT.
You can do it!
The movement of
Let's
genetic
work
material
today
between
to make
organisms
tomorrow a
sideways,
better day!
rather than just
Okay.
vertically from parent to offspring.
You can do
This
it!
speeds up evolution
Let's work
dramatically.
today
The
to make
notes
tomorrow a
mention
better
transduction in
day!
bacteria as a key
Okay.
example. How does
You
that
can
work,
do
and
it.
why is it important?
Transduction happens with bacteriophages, those viruses that infect bacteria. Sometimes when a new phage particle is being assembled inside a bacterium, it accidentally packages a piece of the bacterial DNA instead of, or along with, its own viral genome. When this faulty phage then infects a new bacterium, it injects that piece of DNA from the previous host. Voila.
Genes have been transferred between bacteria that might not be closely related at all. This is hugely important today because it's a major way that things like antibiotic resistance genes spread so rapidly through bacterial populations. A resistance gene can hop from one species to another via a phage.
That's HEGT in bacteria. But the notes make an even more startling claim about our genome, with endogenous retroviruses, ERVs. Right. This is mind-blowing when you first encounter it. ERVs are the fossilized remains of retroviruses that infected the germline cells, the sperm or egg cells of our ancestors millions of years ago. Because the infection was in the germline, the integrated provirus became part of the host's heritable genome. It got passed down through generations, eventually becoming fixed in the species.
And the scale of this,
Yeah.
not trivial, right?
Not at all. The source material states that ERVs and related sequences make up something like 8% of the entire human genome. Think about that. Nearly a tenth of our DNA consists of ancient, inactivated viral code. We are, in a very real sense, part virus.
Wow. Are these just dead relics, or do they do anything? The notes highlight the syncytin gene.
That's the killer example. Syncytin is a gene absolutely essential for the development of the human placenta. Specifically, it helps form the crucial syncytiotropoblast layer, a barrier made of fused cells that controls nutrient exchange and prevents the mother's immune system from attacking the fetus. And where does the syncytin gene come from? It's derived directly from the envelope gene of an ancient retrovirus. Remember, envelope proteins are designed to fuse membranes, viral envelope to cell membrane. Our ancestors co-opted this viral fusion gene and repurposed it to fuse placental cells together to build this vital barrier.
So, complex mammalian pregnancy depends in part on a hijacked piece of viral code.
Essentially, yes. It's perhaps the most stunning example of infection being turned into essential biological innovation.
This kind of horizontal gene transfer, mediated by viruses or similar elements like plasmids, feels almost like it echoes those really fundamental evolutionary events, like how mitochondria and chloroplasts came about.
Absolutely. The endosymbiotic theory, the idea that mitochondria arose from engulfed alpha proteobacteria and chloroplasts from engulfed cyanobacteria, describes ancient, tamed infections that became permanent partnerships. While not identical, the molecular mechanisms involved in integrating and stabilizing foreign DNA, like those used by retroviruses or phages during integration, likely provided the necessary toolkit for these ancient symbiotic events to become permanent. Viral mechanisms may have paved the way for eukaryotic complexity.
But this isn't a one-way street. While viruses are transferring genes, the host immune system is constantly fighting back. This leads to the immune arms race.
It's a relentless battle of adaptation and counteradaptation, playing out over millions of years at the molecular level. Viruses evolve ways to hide, and immune systems evolve ways to find them.
Let's break down some of the viral evasion tactics listed in the notes. What's the most common strategy?
Probably antigenic variation. This is where the virus rapidly changes the bits of itself that the immune system recognizes its surface proteins, or antigens. Think of it like changing disguise constantly. By accumulating mutations, like in HIV, or swapping genetic segments like in influenza, the virus creates new variants that existing antibodies or T cells don't recognize effectively. This forces the immune system to constantly play catch-up.
Okay, changing appearance. What about messing directly with the immune system's communication?
That's immune modulation. A key example is interfering with MHC class I presentation. MHC I molecules are like little display stands on the surface of almost all our cells. They present fragments of proteins from inside the cell to passing immune cells, specifically killer T cells. If a cell is infected, it displays viral protein fragments on MHCI, signaling "I'm infected, kill me." Viruses like CMV and adenovirus have evolved proteins that specifically block MHCI molecules from reaching the cell surface or loading viral peptides. They essentially pull down the alarm flag, making the infected cell invisible to killer T cells.
Hiding the evidence. Clever. What about preventing the cell from just self-destructing? Apoptosis.
Yes, apoptosis inhibition. Apoptosis, or programmed cell death, is a crucial defense. If a cell detects it's infected or dangerously damaged, it can trigger its own destruction to prevent the problem from spreading. Some viruses actively fight this. Poxviruses, for example, produce proteins that mimic the host's own anti-apoptotic proteins, like BCL2. These viral mimics block the self-destruct pathway, keeping the host cell alive and functioning as a virus factory for longer.
And they can even manipulate the broader immune conversation using cytokine mimicry.
Right. Cytokines are signaling molecules that coordinate the immune response. Some cytokines ramp up inflammation and attack, while others dampen it down. EBV, for instance, produces a protein that is a mimic of interleukin-10, IL-10, a key anti-inflammatory cytokine in humans. By releasing this fake calming signal, viral IL-10, EBV can suppress the local immune response, helping the virus establish its infection more easily.
When you combine these evasion tactics with the unique structure of viruses like influenza, you get a recipe for major public health challenges. Let's talk influenza's evolution strategy.
Influenza A is the perfect storm. It has that segmented genome, 8 separate pieces of ssRNA. This structure enables 2 key evolutionary mechanisms. First, antigenic drift. This is the slow, gradual accumulation of small point mutations in the genes for the main surface proteins, hemagglutinin H and neuraminidase N. The viral RNA polymerase is known to be error-prone. It makes mistakes. These small changes mean that the antibodies you developed from last year's flu shot or infection might not recognize this year's strain perfectly. This is why we need updated flu vaccines annually.
Okay, that's the yearly drift, but then there's the big one, antigenic shift.
Right. Antigenic shift is much more dramatic and is what can cause pandemics. It happens because of that segmented genome. Imagine a pig, which can be infected by both human flu strains and bird avian flu strains. If a big cell gets co-infected with both a human flu virus and an avian flu virus simultaneously, as those viruses replicate, their 8 RNA segments can get mixed up and reassorted into new combinations inside the new virus particles. You could end up with a virus that has, say, the hemagglutinin from an avian strain, which humans have never seen before, combined with the neuraminidase and internal genes from a human strain, allowing it to infect humans easily. This radical reassortment creates a completely novel virus subtype. Since nobody has immunity to this new HN combination, it can spread rapidly through the human population, causing a pandemic, like H1N1 in 2009 or the devastating 1918 Spanish flu. So we've seen how viruses challenge us, but that very challenge has forced incredible innovation. As the notes suggest, viruses have unintentionally been our tutors in molecular biology and medicine. Fighting them has given us powerful tools.
Let's look at that toolkit. First, how do we even know a virus is there? Detection and diagnostics. We need ways to find these tiny things, often when they're present in very low amounts.
The workhorse methods are still vital. PCR, polymerase chain reaction, and its quantitative version, qPCR, are incredibly sensitive ways to detect and measure viral nucleic acids, DNA, or RNA. We can find tiny amounts of viral genetic material. This is crucial for diagnosing infections like SARS-CoV-2 or monitoring viral load in chronic infections like HIV. We're directly detecting the virus's blueprint.
So that's looking for the virus genome. What about looking for its proteins or our response?
That's where ELISA, enzyme-linked immunosorbent assay, and Western blot come in. ELISA is great for large-scale screening. It can detect either viral proteins, antigens directly, or, more commonly, the antibodies our immune system produces in response to the infection. It tells you if someone has been exposed. Western blot is often used as a confirmation test. It separates proteins by size first, then uses antibodies to detect specific viral proteins, giving a more definitive result than ELISA alone. Think of the early HIV screening protocols.
And for finding brand new viruses, things we haven't seen before.
That requires broader approaches. Next-generation sequencing, NGS, specifically metagenomics, has revolutionized this. You can take a sample, blood, saliva, tissue, and sequence all the genetic material present. Powerful computer programs then sift through that data, identifying sequences belonging to the host, known bacteria, and potentially novel viruses whose sequences don't match anything in the databases. It's how we identify emerging threats.
And the notes mention using CRISPR for diagnostics. That seems cutting edge.
It is, and it's incredibly elegant. Techniques like Sherlock and Detector leverage the CRISPR-Cas system. They use a guide RNA designed to match a specific sequence in the target virus's RNA or DNA. If that viral sequence is present in the sample, the Cas enzyme binds to it and gets activated. This activation then triggers the enzyme to cut nearby reporter molecules, generating a detectable signal like a color change or fluorescence. It's highly specific, often very fast, and potentially deployable outside traditional labs.
Okay, so we can find them. Now, how do we fight them? Antiviral strategies. Given they use so much of our own machinery, targeting them must be tricky.
Extremely tricky. Unlike antibiotics for bacteria, which can target unique bacterial structures or enzymes, antivirals have fewer unique viral targets. We have to hit the specific steps or enzymes that are viral, not host. That's why understanding the Baltimore classification and the 7-step life cycle is paramount. It tells us what unique viral functions exist.
So what are the main classes of antivirals based on these targets?
Well, thinking about HIV again, we hit it from multiple angles. Reverse transcriptase inhibitors, like AZT, block that crucial retroviral step of making DNA from RNA. Protease inhibitors stop the viral polyprotein from being cleaved into functional units. Integrase inhibitors, like raltegravir, prevent the viral DNA from being inserted into the host genome. Using these in combination, ART is highly effective.
What about other types of viruses?
For influenza, a key drug class targets the release step. Oseltamivir, Tamiflu, is a neuraminidase inhibitor. Neuraminidase is the enzyme flu uses to cut itself free from the cell surface after budding. Blocking it traps the new viruses on the cell surface, preventing spread. We also have broad-spectrum polymerase inhibitors like remdesivir, initially explored for Ebola, then used for COVID-19, which interfere with the viral RNA or DNA polymerase enzyme responsible for copying the genome. These can sometimes work against multiple types of viruses.
It's amazing how detailed knowledge of the viral cycle leads directly to drug targets. But the ultimate twist is using viruses themselves as tools, right? Viruses as molecular tools.
Yes, this is where we've truly turned the tables. We can now engineer viruses for beneficial purposes. Gene therapy is a prime example. We take viruses like AAV, adeno-associated virus, or lentiviruses derived from retroviruses like HIV, strip out their harmful genes, and replace them with a functional copy of a human gene that might be missing or faulty in a patient with a genetic disease. These engineered viral vectors are incredibly efficient at delivering the therapeutic gene into the patient's target cells. They become microscopic delivery trucks for genetic medicine.
And vaccines, of course. Many modern vaccines are virus-based.
Absolutely. From traditional attenuated or inactivated whole viruses to using harmless viral vectors, like in some COVID or Ebola vaccines, to deliver the genetic instructions for a key antigen, to the revolutionary mRNA vaccines, which essentially use our own cells as factories to produce the viral antigen protein, prompting a strong immune response. Viruses taught us how to elicit immunity.
The notes also mention oncolytic viruses for cancer therapy.
That's a very exciting area. The idea is to engineer viruses that selectively replicate in and kill cancer cells while leaving normal cells unharmed. This often works because many cancer cells have defects in their innate antiviral defense pathways, like the interferon response, as part of becoming cancerous. These defects make them vulnerable to viral infection in a way that healthy cells aren't. So the virus becomes a cancer-seeking missile.
It all comes full circle. And the ultimate irony, connecting back to the arms race, is the origin of CRISPR.
It really is profound. CRISPR-Cas9, this revolutionary tool allowing precise gene editing, wasn't invented in a human lab from scratch. It was discovered by studying how bacteria defend themselves against bacteriophages. The CRISPR system in bacteria is essentially an adaptive immune system. It captures snippets of DNA from invading phages and stores them in its own genome, the CRISPR array. If that phage attacks again, the bacterium uses RNA copies of those stored snippets, guide RNAs, to recognize the phage DNA and directs the Cas enzyme to cut and destroy it. So the foundation of arguably the most powerful biotechnology of our time is literally a bacterial defense mechanism against viruses. Our greatest tool for manipulating genomes came from studying how microbes survive viral attacks.
We've really journeyed through the intricate world of viruses, structure, hijack, latency, evolution, and now even harnessing them. Let's try to synthesize this, anchoring it with some key clinical takeaways from the notes.
Sounds good. The story of HIV, a class 6 retrovirus, highlights latency via integration and the triumph of multi-target art therapy. It showed us that chronic viral infections can be managed by hitting multiple unique viral steps at once.
And HPV types 16 and 18 showed the direct link between viral proteins, E67, disabling tumor suppressors, P53RB, and causing cancer, but also the power of prevention through vaccination, like with Gardasil.
Exactly. Influenza A remains our annual reminder of antigenic drift and the ever-present threat of pandemic shift due to reassortment. SARS-CoV-2, a class 4 plus ssRNA virus, became a global crash course in virology and demonstrated the incredible speed possible with new diagnostic and mRNA vaccine technologies.
And we shouldn't forget the historic victories, like the near eradication of poliovirus, another class 4 virus, thanks to effective global vaccination campaigns using tools like the inactivated vaccine, IPV.
Each of these cases reinforces the central theme. Understanding the fundamental molecular biology of the virus,
Let's work
its
today to
Baltimore
make
class, its
tomorrow a
replication
better
strategy, its
day.
unique enzymes is
Okay.
absolutely key to
You can
developing
do
effective
it.
diagnostics,
Let's work
therapies,
today to make
and
tomorrow
vaccines.
a better
But
day.
the legacy, as we discussed,
Okay.
goes so much deeper than
You
just
can
disease.
do it.
Let's revisit
wo-
that idea of mitochondrial and plastid echoes.
Right. The theory of endosymbiosis posits that these crucial eukaryotic organelles originated from bacteria being engulfed and forming a permanent symbiotic relationship. It was, in essence, a tamed infection that became indispensable.
And the source notes propose that the kinds of molecular tools viruses possess mechanisms for DNA integration, for horizontal gene transfer, for managing foreign genetic material might have been crucial facilitators in making those ancient symbioses stable and heritable.
It's a compelling idea. Viruses and related mobile genetic elements provided a pre-existing toolkit for cutting, pasting, integrating, and regulating DNA. This toolkit could well have been co-opted or utilized during the complex process of merging whole bacterial genomes into an early eukaryotic host, stabilizing the partnership. The viral world may have provided the molecular glue for eukaryotic complexity.
This deep historical and molecular context really underscores the importance of the scientists who pieced this puzzle together.
Absolutely. We stand on the shoulders of giants. Temin and Baltimore winning the Nobel in 1975 for discovering reverse transcriptase fundamentally changed our understanding of information flow in biology. Harold Zerhausen made the crucial Nobel-winning link between persistent HPV infection and cervical cancer. Luc Montagnier and his team's discovery of HIV opened the door to understanding AIDS.
And bringing it right up to date, Doudna and Charpentier's Nobel for CRISPR, recognizing that this revolutionary technology originated as a bacterial defense against viruses.
It all weaves together. The study of viral infection isn't just about disease. It's about understanding the fundamental mechanisms of biological programming, reprogramming, immunity, gene regulation, evolution, and even the origins of biological complexity itself. The logic is interwoven. #tag outro.
Which brings us back to that central idea from our source material. Viruses as molecular poets of existence. Yes, they are parasites, absolutely, exploiting cellular machinery for their own replication. But through that process, as we've explored following these detailed notes, they drive the immune arms race. They literally write themselves into our DNA as ERVs, becoming part of our inheritance. They even seem to have spurred the evolution of chromatin regulation, things like DNA methylation, that our cells use partly as an epigenetic defense to silence those ancient invading viral sequences. They force adaptation, turn infection into inheritance, chaos into code. And that leads us to a final thought prompted by this deep dive into the material. If 8% of our genome is recognizable ancient viral debris, and we see examples like syncytin, where viral genes were co-opted for crucial functions, what other parts of our biology, what other regulatory networks, maybe even some of that mysterious junk DNA, are actually silent echoes, the still functional or repurposed legacies of countless other viral encounters deep in our evolutionary past that we just haven't deciphered yet? The full extent of their influence is likely still hidden within our own code.